Welcome to our lecture 25. This is part 7 of the chemistry of microbiology, which we are talking about proteins as part of organic macromolecules. Kindly contact our contacts below for any information. You are advised to subscribe to our channel to stay tuned. Proteins. They are organic molecules that are mainly composed of carbon, hydrogen, oxygen, nitrogen, and sulfur. They perform numerous functions depending on its shape and in the cell. Such include structural functions. There are structural components in cell walls, membranes, and within the cells themselves. They are also the primary structural materials of the flagella and cilia. Those are the movement elements of microorganisms. They are part of the hair, nail, outer cells of the skin, and muscles. They perform enzymatic activities. For example, enzymes are proteins, catalysts, which enhance the speed or likelihood of chemical reactions. Regulation. Some proteins, like hormones, regulate the cell function by stimulating or hindering either the action of other proteins or the expression of genes. In transportation, some proteins act as channels and bumps that move into and out of the cells. They work in defense and offense. Some bacteria produce proteins called bacteriosis that kill other bacteria. Antibiotics and complement are examples of proteins that protect the body against microorganisms. Amino acids. Proteins are polymers composed of monomers referred to as amino acids. Amino acids contain basic amino groups, a hydrogen atom, a carboxylic group, and a side group are all attached at the same carbon atom. That's alpha carbon. The side groups may be a single hydrogen atom, various chains, or complex ring structures. There exist numerous amino acids, but only 21 of them are used by organisms in synthesizing proteins. The side groups affect the way amino acids interact with another within a given protein or how a protein interacts with other molecules. The amino acids are soluble in water because they contain both an acidic carboxylic group and a basic amino group, thus posing both positive and negative charges, making them easily dissolved in water. Aqueous solutions of amino acids and simple sugars blend light rays, passing through the solution either clockwise, that is D forms, or anti-clockwise, L forms. The below the mirror images of the D and the L forms. Many organic molecules exist as D and L forms that are stereoisomers of one another. Either they have the same atoms and a functional group but are mirror images of each other. Most amino acids in proteins are L forms, except for glycine that doesn't have a stereoisomer. Most organisms almost always use D sugars such as D-glucose in metabolism and polysaccharides. Some of the amino acids found in human proteins include glycine, alanine, valine, leucine, isoleucine, serine, threonine, cytosine, methionine, glutamic acid, aspartic acid, lysine, arginine, asparagine, glutamine, phenylamine, tyrosine, histidine, tryptophan, and proline, among others. Peptide bonds. Amino acids in the cells are linked together by a dehydration synthesis reaction that leads to formation of a covalent bond between the carbon of the carboxylic group of the one amino acid and the nitrogen of the ammonium group of the next amino acid in the chain. The covalent bonds between the amino acids are named peptide bonds. A molecule composed of two amino acids linked together by a single peptide bond is called dipeptide, and longer chains of amino acids are called polypeptides. Proteins are unbranched polypeptides made up of hundreds to thousands of amino acids linked together. The peptide bonds in a certain pattern as determined by genes. The specific protein structure determines the functions. Proteins have at least three levels of structure, that is, primary structure, secondary structure, tertiary structure, and quaternary structures. Primary structure. The primary structure is basically its sequence of amino acid. It varies in length and amino acid sequence. A change in amino acid in the structure can drastically affect a protein's overall structure and functions. For example, the replacement of amino acid valine by alanine in position 136 of the primary structure of a particular sheep-brain protein called cellular protein may result in a disease called scrapie. The altered protein spread into cows cause mad cow disease, and into humans cause variant Creutzfeldt-Jacobs disease. Although numerous substitutions can be made in some uncritical regions of cellular protein with no negative effect. Primary structure. Secondary structure. Ionic bonds, hydrogen bonds, hydrophilic and hydrophobic characteristics cause many peptide chains to fold into coils, alpha helix or accordion-like structure, thus B-splitted sheets. Proteins are typically composed of both alpha helix and beta-splitted sheets linked by short sequence of amino acids that do not show such secondary structures. Because of its primary structure, the protein that causes variant Creutzfeldt-Jacobs disease has beta-pleated sheets in locations where the normal protein has a alpha helix. The structure of secondary protein is as below, that's showing the alpha helix and the beta-pleated sheets. Tertiary protein structure. Polypeptides further fold into complex three-dimensional shapes that are not repetitive like alpha helix and beta-pleated sheets, but are uniquely designed to accomplish the functions of the specific protein. Some proteins form strong covalent bonds between sulfur atoms of cysteine molecules that are brought into proximity by the folding of the polypeptides. These disulfide bridges help in maintaining the tertiary structure of many proteins. Quaternary structure of proteins. Some proteins are composed of two or more polypeptide chains linked together by disulfide bridges or other bonds. The overall shape of such proteins may be globular or fibrous. The tertiary structure and quaternary structure are as shown in the image. Organisms may further modify the proteins by combining them with other organic or inorganic molecules. For example, glycoproteins are proteins covalently bonded with carbohydrates. Lipoproteins are proteins bonded with lipids. Metalloproteins contain metallic ions, and nucleoproteins are proteins bonded with nucleic acids. Apart from substitution of amino acids in the structure of proteins, other physical and chemical properties such as heat or temperature, changes in pH, and salt concentration may interfere with hydrogen and ionic bonding, which in turn disrupt the three-dimensional structure of proteins. This disruption is called denaturation, which may be permanent or temporary. Thank you for viewing our lecture. Kindly subscribe for more.